Hi guys, how are you doing? I hope you're doing very nice and you enjoyed our previous videos and the lectures. And today we will talk about our next tutorial about the title of the tutorial is about how to identify a research target, a research topic to study. And today we have another team member who will help us to design this and who will ask a question on your behalf. We are trying to she will ask us question that probably most of you guys have in your mind, and so welcome her, and I hope you will like the video.
So what this video is all about?
In this video I want to tell you what people do study in the lab, what kind of problems they choose, how they find which problems or which question they should address, and how they decide what they want to work on, which kind of area or what things they work on. And I understand that this might be quite confusing for you at the very beginning, but I'm pretty sure that at the end of this video you will understand what I'm talking about.
Okay, so you want to say that if we choose a question, a biological question, so you are going to describe that how we start working on that question in lab.
Exactly, very true.
Okay, so just tell us how you start.
So again, I will tell you then, if you want to study something, the important question is always for you that where to start. That's I think for question to everybody for any area. Even if you want to go for traveling, you always start thinking that, okay, I want to do some packing, but where should I start? What should I pack first and what should I pack last? And the same kind of analogy you can put in a research work also. So before you enter in any research project, you actually have to have very clear idea that from where you want to start. So I will give you an example about these things, and then you probably will understand what I'm talking about. So let's talk about a cell. So we know that a cell, then there is a cell in the nucleus, this is a cytoplasm, and we know that there are a lot of cellular activities that are going on in the cytoplasm. Then there are some other neighboring cells which are communicating with this cell that we are studying, right? And this kind of some kind of communication is going on in the middle, and a lot of activities going on in cytoplasm, and a lot of activities are going on in the nucleus. So you can see this is a really very complicated process, and these processes are carried out are governed by the different biomolecule. For example, we know that the DNA, we know the RNA, and we know the proteins, right? And so right now I will not tell you very detail about it here, but what you see here is that all kind of cellular activities are not just a unidirectional process, but is a kind of connected network. So one biological process is actually governed by facilitated by a lot of proteins, a lot of RNA, and somehow involve the DNA in the whole process. So it's kind of adding more complexity, right? And we also know that in some situation these normal cells can also get a disease, for example cancer, right? And when the cancer state comes, it usually comes that when some kind of normal biological process which are running very smoothly are altered because of some other elements or factors. So let's say there's a protein X which is now functioning in cell-cell interaction here, but due to some mutation this X protein is converted into X prime. So, and now this mutant protein cannot function anymore, so the cell-cell interaction now lost, and now this cell is in danger. So either it will be degraded, it will be destroyed by apoptosis or any means. So here you can understand that one single protein here, a little mutation in this protein actually affect the whole cellular processes at all. So you can see that these are very, very complex problem, and you cannot study all the complex system at once. So you cannot study all things, whatever is going on in the cell at once. So you have to choose one target. What you want to study. Either you want to work on a protein level, you want to understand that what kind of proteins involved in cancer, how many proteins are there, which protein is playing what function, and what happened if the some protein stopped functioning at certain point of the time. So this is a kind of picture that I want you to have in your mind. So there are different biomolecules in a cell which which perform functions as they are designated for, and this is we want to identify that if we are provided with a patient or we are provided with cells which has cancer, what we can study, how we can choose, and etc. Yeah.
Okay, so here you want to say that any alteration in these biomolecules cause a particular disease.
Indeed, yes.
Okay, so can you just tell us that with an example that how we can choose that what we want to work on?
Well, that's a very difficult question actually. So what we want to work on uh, is depends on the knowledge what we have. So for example, there are uh, three biomolecules which I just introduced you. Maybe I can give you a brief introduction about these things, DNA, RNA, and protein, and then maybe we can proceed with what kind of research you want to opt for. What do you say?
Okay, suppose we can start with proteins.
Okay, so proteins. Why the proteins are so important? So the question is why the proteins are so important, right? Um, if we remember correctly, we are studying about the protein since our school days, right? And we have studied one line is that proteins are the building block of cell. What does that mean? So the building block means the proteins are something which actually give a shape to a functional cell, and the protein can play a different kind of roles. For example, they can play a structural role. They also perform function. So what the function means? Function means they perform enzymatic activity, they regulate the processes, um, they govern some function. For example, ion channels that we know, so the ion channels are also protein. For example, sodium potassium pump that you're very familiar with, right? And sodium potassium pump is also a protein channel, but that's also a protein, right? That's also a functional protein. What about the structural protein? How we can say that this protein is structural? Um, the very fine example of the structural protein is the keratin.
Mm-hmm.
Um, so proteins are actually divided in a vast range. So, I would definitely tell you about the different kind of protein. For example, structural protein. There are functional proteins. Um, there are also proteins which do not do anything, and we call them scaffold protein. Okay. So about the structural proteins is about how they shape a structure of a cell. So we know that there are several proteins which present in a cell membrane, and they perform interaction with other cell in the same environment, right? So cell-cell interaction. So the protein involved in cellular interaction. Other kind of structural proteins is like keratin, I told you. Yeah. What about the functional protein? So actually, if you look at them, the functional protein, actually, there are a lot of functional proteins. There are a few examples which I will tell you about is that um, sodium channels, sodium ion channels. And the other fine example is about G protein coupled receptors. You probably have heard about GPCRs, G protein coupled receptors. The other functional proteins are enzymes like hydrolase, oxidoreductase, maybe very familiar one that you know already is amylase. So these are the functional proteins, and the scaffold proteins are generally present in DNA. So these proteins usually help to shape a structure of DNA in the form of chromosomes. So these are targeted to DNA or sometime between two cells. So it's like this. All they are also filling the gap between the gap, the empty space between two cells or three cells. Right? And what you know about this? So what we actually now know that these proteins have a certain specific role inside the cell. Doesn't mean that the structural protein are not important, right? Because if they are performing some kind of cellular interaction, then they are important. If those protein will not be there, the cellular interaction will not be facilitated, and this will affect the whole biological system, right? And the same is true for the functional protein. For example, if the GPCR will stop functioning, then you will get a lot of diseases, because the GPCR, the G protein coupled receptors, are the very important proteins which facilitate the cellular signaling, right? For example, the insulin thing, I mean, there are other receptors, but there are different hormones that you know, and how these hormones are functioning is through the GPCR. So now we understand that these proteins are very important to perform any cellular function, right? And what happens if we simply take out a protein from the pool? So for example, these total protein, which are just a rough example, there are total 100 proteins, okay? And out of these 100, of course, you cannot study all the 100 protein together, right? So you have to understand, you have to identify which protein you want to study. And how we will actually identify which protein we want to study is there are different methods to look at it, which you want to study. So the one method is to look at the literature. So the literature means published research papers. For example, a research paper is talking about a protein M, okay? And he actually characterized the protein M in his research paper. And at the end of the paper, he's talking about that the protein M is interacting with, is found to be interacted actually. So they actually discard as the is found to be interacted with protein N, and they have not characterized this protein N. So this may be the very new finding for them, and while they were studying the protein M, they do not want to spend more time to characterize the protein N, right? Because they do not want to spend more time, they want to publish whatever they have got. So what you can now identify, okay, so you are studying a paper which is recently published in 2016, and you found that the protein N is a very new protein, and what you can do now, you can propose to the lab, okay, you want to characterize protein N, you want to work on protein N, because that's very new protein, it's found to be interacting with M, that means it may have some functional role, or it's affecting the function of protein M. Great. This is the literature way that you actually find out the protein about the proteins.
So here you mean to say that this can be our new question, new biological question which we can solve.
Exactly. So that's how we identified a very new biological question from the literature, that which protein we want to study.
Okay.
I mean, this is just one example through literature. There are another example that I definitely want to tell you about is so this is literature from the paper, and the other example is just give me a second. Okay, so the other thing which you can study, for example, you are just doing your masters, and somehow you got a chance to work as a summer intern in a lab in India or anywhere, right? And you go to the lab, and then what your purpose is to learn some experimental thing you want to you want to study the lab work, right? So you go to the lab, you talk to the supervisor, and your supervisor tell, okay, we are working on keratin protein. We are studying how these proteins are folding, and what happened to their folding, how they unfold, how they misfold. I mean, what kind of, so they are studying the protein folding problem. Okay, so they are studying a protein folding problem there. So you go to the lab, you talk about you talked about the professor, and your professor will tell about, you know what, we have a similar protein to the keratin, and we recently got this protein and we are studying, would you be interested to work on this protein? So you identified it very right. This is the second way of identifying the target or choosing the target. So this target is told to you by your supervisor. So your supervisor said that you have to work on this protein because we are interested in this, right? So this is the second way, and this usually happen in developing countries because uh, they already have some kind of interest which they are working in the lab, their research interest, and when you go join their lab for summer training or for PhD, they used to give you the topic or title which they want you to pursue. So this is the second way of identifying the target. The third way of identifying the target is the kind of, people say it's computational analysis, like using the tools of bioinformatics. So our third target could be using bioinformatic analysis. And I will tell you, this is another very common way to identify the target and how we will do this, right? Okay, so we know that there are several bioinformatic programs which can tell us about proteins, about different proteins in the different organisms, right? Okay. Generally, you probably have heard that people study the bacterial proteins, right? So, for example, there is a protein called ATPase or ATP synthase. And this is a very large multimeric protein complex which is present in bacteria, and it helps the cell to generate the ATP. And we know that the ATP is very important molecule for the cell. It's kind of energy currency, right? So the ATP synthase is a very important complex, and it is present from bacteria to humans. That means from prokaryotes to higher eukaryotes. It's like it's present everywhere. Just imagine a situation that in 1970s, a lab resolved the structure of a bacterial ATP synthase. Okay, but they do not know how the ATP synthase look like in human, and what are the proteins. Does the actually human have those protein which the bacterial ATP synthase complex has or not? So this is a situation, right? And now we know that the ATP synthase complex, for example, complex has roughly 30 proteins in bacteria. Another study in another part of the world identified not the structure of the human ATP synthase, but they identified that there are 35 proteins in the human ATP synthase complex, right? So now you identified there are 5 new proteins here. I mean 5 more proteins, so we don't know how many new proteins are there, right? So here we will take the advantage of bioinformatic analysis. We know the 30 proteins here, we know those protein sequences, and we know what subunits they are, right? Now what we will do, we will take one by one protein, and we will do a computational analysis to identify whether a protein X, which is present in bacteria, is there any homologous protein which is also called X prime, is present in eukaryote or not. Okay, So doing this, I mean, you will do this using a BLAST algorithm, a BLAST program. So if you don't know, don't worry. We will tell you about how to use the bioinformatic tools and methods to identify new targets. But here it's just to explain you how things how things goes on. Okay. So, and by doing this, you analyze all the 30 proteins. What you found that only 25 proteins are present in both bacteria and in human, but there are 5 proteins which are present in bacteria, but they are not present in human. Okay, and you also know that there are some more proteins in the eukaryotic system. So now you know at least you have 10 proteins that are very new proteins. People have not studied them, they have not characterized them, and that's how you can choose a protein from these 10 protein pool, and then you can study. You can propose that, okay, this is the protein which is present in bacteria, but it is not identified in human. What protein in human is functioning at this place? So this is another way of identifying a target.
Okay. So these are the ways of identifying the targets for the kind of questions we deal with in biology.
Yeah.
And then further, it's like when we start doing research on them, then we need to actually kind of fragment our problems in this way.
Yeah.
Okay.